Nithya Ganeshan from 3V College. I am an assistant professor in biochemistry. Today we are going to see about the key terms which are used in developmental biology. Okay, so let us start. So what is developmental biology? It is the study of how organisms grow from a single cell into a complex organism. For example, a plant which grows from a seed or a baby which grows from a zygote. The first term is embryology. Okay, embryology refers to the growth of a cell into an embryo from the gametes. Okay, so the gametes are nothing but the egg and the sperm which unite to form the zygote, which is the fertilized egg. So as we can see in this diagram over here, this is the sperm and the egg which meet to fertilize. It forms the zygote, and the single cell zygote, it divides, and the division is known as cleavage. Okay, so first one cell will divide to form two cells will divide to form 4 cells, and so on. Okay, so it reaches the 4 cell stage, and once it reaches the 16 cell stage, it is called a morula. So morula is a solid ball of cells. It looks like a mulberry. Okay, mulberry berries. And once the morula is formed, further division, it forms a hollow ball. Okay, and this is known as the blastula. So blastula, it is hollow in the center, and the cells in the center will form the embryo. Okay, so this is embryology, which is the study of formation of an embryo by fusion of gametes, the egg and the sperm. Then another important term which is used in developmental biology is potency. Potency is ability of a cell to differentiate into other cell types. So one cell can form many different roles. Okay, and we have different types of potency. Mostly when we refer to potency, it is about the stem cell. Okay, stem cells are cells which can form the various cells in the body. Okay, so the different types of potency. First we say totipotent. Okay, totipotent, the cell, it can differentiate into any cell. For example, the cells present in the 16 stage cell morula, each cell, it can give rise to any cell in the body, even the placenta. Okay, but most of the cells, stem cells included, they are always pluripotent. Other than the cells of the morula, they are pluripotent, which means they can form any cell type except the placenta. Okay, for example, cells in the blastocyst, from the inner side of the blastocyst, they can form the embryo, but they cannot form the placenta. Okay, only the placenta cannot be formed. They are said to be pluripotent. Then the next type of potency is multipotent. Okay, it can form a set of closely related cells. The best example is the hematopoietic stem cell. Okay, it can form only blood cells, but there are many blood cells. It can form white blood cells, red blood cells, it can form the T cells, B cells, macrophage, it can form the platelets. Okay, so it is said to be multipotent. So it can form several cells but are of a single related cells. Okay, next type we have oligopotent cells. It can form specific cell types only. For example, in hematopoiesis, we have the hematopoietic stem cell. From that, we get the lymphoid progenitor cell. So these lymphoid stem cells, they can only form the B cell or the T cell, the T lymphocytes. They cannot form the red blood cells. Okay, so they can form only a specific number of cells. So they are said to be oligo. Oligo means a small number. Okay, and next type of potency is unipotent, which means it can form uni meaning one. It can form only one type of tissue. Okay, for example, epidermal stem cells, they can form only the skin cells, epidermal cells, they can differentiate to form epidermal cells. Okay, and there's another last term which is nullipotent. That means it cannot differentiate at all. For example, the red blood cells in the body, they cannot differentiate, they cannot form any cells. Okay, they, after 120 days, the red blood cells, they are renewed by the body. Okay, so that is nullipotent. So we see potency, ability to differentiate into other cell types, and the different types. Totipotent, it can form any cell. Pluripotent, it can form the embryo, any cell other than the placenta. Multipotent, it can form a group of closely related cells like blood cells Oligopotent, it can form only a few cells like the lymphoid progenitor forming only the lymphocytes. Unipotent, it can form only one type of cell. For example, epidermal stem cells can form only the skin cells, and nullipotent, which means it cannot differentiate at all, like the RBC. Okay. And then another term, few terms we are going to see in the different stages of commitment. So first we see what is commitment. Commitment means the fate of the cell is decided. Okay, so some cells will become the nerve tissue, some cells will become the heart tissue. So what is the cell going to become? That is the commitment of the cell. Okay, so commitment means the fate of the cell, it is decided. So what are the different stages of commitment? First we have naive cells. So naive, in English the meaning is innocent. So naive means it's an innocent cell. For example, a hematopoietic stem cell. Okay, is an or the naive T cell, B cell. So it has not encountered an antigen, it is called a naive cell. So a naive cell means it has not yet differentiated, it can form any other cell. Okay, it can differentiate to form any other cell. Then the next stage of commitment is specified cell. Okay, so specified cell, how does a cell become specified? It is by cytoplasmic determinants. So some cytoplasmic determinants, they make a naive cell into a specified cell. So again, this term we need to understand what is cytoplasmic determinants. These are just chemical signals which are present in the cell. So cytoplasm, it is present in the cytoplasm of the cell. These chemicals, they give a signal to the cell to differentiate. Okay, so it becomes a specified cell, which means it is in the process of becoming another cell. Okay, it is in the process of forming a tissue. A specified cell, it can be reversible. It can reverse back into a naive cell. Okay, and it can form something else also. So that is why it is said to be reversible process, formation of the specified cell. Okay, for example, the hematopoietic stem cell will form the myeloid progenitor cell. So this myeloid progenitor, what it is it can form only the lymphocytes, or it can form the blood cells, red blood cells. Okay, it cannot form the lymphocytes. So this is a specified cell. Then from this we go to the next stage of commitment, which is a determined cell. So once a specified cell becomes a determined cell, its fate has been determined. That means it cannot be reversed. So a determined cell, for example, megakaryocyte, it cannot form anything else. Okay. So it is said to be, the stage is said to be reversible. So when will a cell become determined? When it loses its competence. Okay. So the loss of competence leads to formation of a determined cell. Okay. So that means competence means the ability of the cell to differentiate. So once the cell has lost its ability to differentiate, it becomes a determined cell. Okay. And the last stage of commitment we see is a differentiated cell, or the last stage, the cell, it becomes functional. Okay, it gets its specific function. And how this takes place is by cell specific gene expression. Okay, so the cell, it has become differentiated. So once again we see these are the different terms which are being used in developmental biology. Commitment meaning the cell is, fate is being decided. So what is the cell going to become? It is going to become that. Okay, a naive cell is a cell which is not yet committed. Okay, and in the next stage of commitment we have the specified cell. So a specified cell, it's in the process of becoming committed, so it can reverse back. And then the third stage is the determined cell, which means the process is irreversible. Once it becomes determined, it cannot reverse back. Okay, and a differentiated cell is the final stage. So the cell is completely differentiated, it receives its function and it becomes a fully functional cell, like a nerve cell. Okay, so once it is formed, it is that cell. So next we see what is fate map. Okay, so this is another commonly used term in developmental biology. So we saw the cell fate is decided. So supposing this is the embryo. Okay, this whole thing is the embryo, and each group of cells in the embryo, they have a specific fate. Okay, so a fate map, it's nothing but a diagram which shows which parts of the embryo will develop into adult tissues and structures. Okay. For example, in this picture over here, this part is going to form the epidermis. This part is going to form the neural tissue. Okay. This is going to form the notochord. So, and this lower portion, it's going to form the endoderm. So, this is the fate map of a frog. So, the cells present in the blastocyst, we decide the fate. Okay. So, it's a diagram which shows the parts of the embryo which will develop into adult tissues and structures. So, which part is going to form the head, which part is going to form the tail, which part is going to form the notochord. All this is decided in the fate map. So, cells present in that area, their fate is to become a part of that structure. Okay. Okay, next we are going to see what is a morphogenetic gradient. First, we see what is morphogen. Morphogen, they are nothing but chemical signals within the cell. Okay, they are a set of chemical substances. They give a signal to the cell. So, these morphogens, they arrange themselves in the form of a gradient. Okay, to decide the fate of the cell. For example, in this picture, we have two morphogens, SHH and BMP, that is the bone morphogenic protein and the sonic hedgehog protein. So if you see this diagram over here, SHH is maximum on this side and it is gradually reducing, and on this end, on the dorsal end, BMP is maximum and its concentration is minimum on the ventral side. So according to that, the fate of the embryo is decided. Okay, so the fate of the embryo according to the morphogens, how they are arranged. So these chemicals, they are arranged in a gradient. This is known as the morphogenetic gradient. Okay, and this decides the fate of the embryo. So which part of the embryo is going to form the head, which part is going to form the tail, which part is going to form the ventral side, the dorsal side, the front, the back, all this, it is decided by the chemicals which are present within the cells. Okay, and these chemical substances are called the morphogens. They decide the fate of the cells. So this is known as a morphogenetic gradient. And the last few terms over here, we know genomic equivalence. Genomic equivalence means all the cells of an organism, they have the same genetic information. This is known as genomic equivalence. For example, if it's a human being, all the cells will have 23 pairs of chromosomes and the DNA will be similar for the same organism. Yes. So that is genomic equivalence. Okay. A theory that the cells of an organism contain the same genetic material. So again, next term is mutant. Mutant is an organism whose structure is changed because of a change in the genes. For example, if you see this picture, normal snake has one head, but this is a mutant. It has two heads because of some change in its genetic structure. Okay, so change in genes which leads to structural changes is known as the mutant. Okay, and then next we see imprinting. So imprinting, this is a quality of animals. It is the learning. For example, if if the chicken egg is hatched by a duck, okay, and the chick once it comes out, it will think of the duck as its mother and it will get attached to the duck. Okay, so that is imprinting. So some behavior in animals, it is learned. Okay, that is known as imprinting. And the last term we will be seeing is transgenic analysis. So what is transgenics? Transfer of the gene. Okay, so gene from one organism, it is transferred into another organism. This is transgenics. So we are able to even make different animals in transgenics. Okay, so why is transgenics used in developmental biology? It is used to study the effect of a gene. For example, if you insert a specific gene into the animal, what is the changes that it brings about? So we know this is the effect of this gene. Okay, so how do we do transgenics? You take the fertilized egg of the mouse. In the blastocyst stage, you take out a few cells and you insert cells which have been manipulated or in which the gene transfer has been done. So those are inserted into the blastocyst stage, and once it develops, it's in the blastocyst, it's implanted into a surrogate mother, and once the baby develops, The offspring, it's said to be a chimera. Chimera means it is a hybrid. Okay, it is a mixture of the genes from the rat and the genes which have been manipulated. So if you see, they have drawn like a mosaic pattern. So instead of developing into a normal mouse, it will develop into a transgenic animal. Okay, and this it can be used to study maybe the effect of insulin or whatever it is we want to study. Okay, so to study the effect of a gene, insertions have been made, okay, in the gene. So with this we finished learning about the key terms in developmental biology. I hope you enjoyed this. Thank you. So again, this is a mutant of many insects together. Okay, thank you.